So in the next part we'll look at a group known as non-nutritive components, which we'll discuss in a moment. We've sort of talked about one already in terms of vitamin B12. We'll look at some others here and look in particular at vitamin A. So non-nutritive components. These are often described as components compounds that do not provide energy, for example water and fibre. Also colour and flavour compounds. This broad definition would include vitamins and minerals and many phytochemicals. The latter are chemicals produced by plants which may affect health but are not, generally speaking, seen as essential nutrients like vitamins. This topic we covered later in this and other modules, particularly in the section on functional foods. We'll look at one example of a metabolism in detail, that of vitamin A. The metabolism of alcohol will be discussed in the next lecture in some detail, as it is non-nutritive in the broadest sense, but apart from other issues, is a significant contributor of energy for various people. So it's a bit of an overlap in definitions there, I'm afraid. Okay, we'll start by looking at metabolism of vitamin A. There will be a little overlap with one of the micronutrients lectures, but that is clarity is appropriate here. Right, so sources of vitamin A, as well as sources of vitamin A such as liver, fish, oils, milk and eggs. It is nutritionally available from a range of vegetable sources as what are called vitamin A active compounds. All trans retinol, as shown there, is vitamin A. Related compounds such as 11-cis retinaldehyde and 9-cis retinoic acid can be metabolized to vitamin A. The conversion is not complete, but such molecules may be nutritionally important sources of vitamin A. In most textbooks and papers you will see retinol drawn like this. It may seem a little bit confusing until you realise that most of the carbon and hydrogen atoms are assumed. In fact, I couldn't find an online image with all of the atoms, so I had to draw this myself. This one, there it is. As you'll see, the former depiction is much clearer, and this is even more so true for bigger molecules. Referring back to biochemistry, although we will refer to retinol or vitamin A from now on, it is worth reminding ourselves of the cis and trans nomenclature of double bonds. The 3D nature of biological molecules is key to their function. The cis version of retinol would not be biologically active since relevant enzymes could not act on it. Similarly for 11-cis retinaldehyde, note that when you are doing this in reports, cis and trans are written in italics. Okay, beta-carotene. Beta-carotene is the most important nutritional source of vitamin A. We've already sort of looked at it in other lectures in terms of its role in nutrition and as a food colour. Vitamin A activity is expressed as a proportion of the activity of retinol, as I say, vitamin A itself. This is much higher since retinol is easily absorbed and metabolized, whereas provitamin A compounds like beta-carotene require significantly more metabolic processing, so less biologically available. Again, we reviewed this in the other lecture. So fat solubility. There's a principle in chemistry that like dissolves like. That is, compounds with similar chemical structures will tend to be soluble in each other. A characteristic of fats and fatty acids is that they have a long chain of carbon atoms. Similar structures are also found in carotenoids like beta-carotene and molecules like retinol, which is characterized as a fat-soluble vitamin, which among other things has important implications for its metabolism, transport and storage. We can store fat-soluble vitamins, unlike water-soluble ones like vitamin C. Okay, from food to retinol. The overall process is complex, and we are summarising here somewhat. Vitamin A and beta-carotenes are typically enclosed in plant cells, which have relatively strong walls compared to animal cells. Unless these cell walls are broken down, for example by cooking and chewing, these nutrients may remain biologically unavailable, so that's quite important. This process here starts in the stomach and continues in the small intestine, eventually releasing retinol. Bowel salts again. Bowel salts, as mentioned earlier, have a similar structure to detergents, and like them are surface active agents. That is to say, molecules that are parts that are soluble in water and parts that are soluble in fat. They form micelles with lipids and lipid-soluble compounds like beta-carotene and vitamin A. This allows transport of these fat-soluble molecules and promotes their metabolism. An example of a relevant bile salt here is torricellic acid, as shown. Before we look at the process of transporting vitamin A around the body, we should discuss how beta-carotene is converted to retinol. This takes place mostly in the micelles previously described and is catalyzed by enzymes such as beta-carotene deoxygenase, with bile salts stimulating the enzyme action. This is the basic chemistry. First, beta-carotene is hydrolyzed into two molecules of retinol by one enzyme, and then each of these molecules is converted to retinol by another enzyme. Okay, transport and use. Again, it's a reasonably complex process which we are summarising here. Stomach action will have released both vitamin A and carotenoids from the food matrix. They then move down into the small intestine and aggregate with lipids and bile salts to form micelles. Remember, they are fat solubles. And again, in the small intestine, the micelles are hydrolyzed with pancreatic lipase, releasing retinol, which is then absorbed by the mucosal cells of the small intestine. The retin is packaged into something we've seen before, that is to say silomicrons, and these are used for transport around the body. The conversions of provitamins to retinol again also takes place in the small intestine. We discussed silomicrons briefly before. They are have the heaviest mass of any lipid-containing particle in the body, and are the richest in triglycerides, that is to say simple fats. They transport dietary fats from the gut to the adipose tissue, where it may be stored. Liver and muscle cells are also involved in transport of retinol to tissues. They are transported initially via the lymphatic system into the general circulation. In the bloodstream, they are transformed into silomicron remnants by hydrolysis of the core triglycerides by lipoprotein lipase, releasing their contents for use in tissues. Okay, vitamin A and vision, which is one of the key rules of vitamin A. This is the visual cycle. It is a cycle. It involves initially the conversion of 11-cis isomer of retinol into the all-trans isomer, a process which requires the protein opsin and light. The reaction then reverses and starts again. These are the two retinol isomers. Note the kink in the overall shape produced by the 11-cis bond. The opsins are said to be G-protein couple receptors. G-protein couple receptors are found all over biology, in things like taste buds, for example, for sweetness and umami, the taste of proteins. They're made light sensitive by what's called a chromophore, such as in this case retinol. They're aggregated in the discs into the vision system, as shown here. And there's a brief summary of the process as well. Reaction to light. The reaction of reduction to light is very rapid, of the order of picoseconds, a picosecond being 10 to the minus 12 seconds. It's the only light-dependent step in the vision process, and it's sometimes called bleaching for the reason we'll discuss in the next slide. Recovery, particularly from a bright light, is a much slower process than activation by light. That's why it takes a significant time for your eyes to recover after looking into a bright light. You still see after images for quite a while. Just to finish off this little section, a summary of some definitions and issues. Dark adaptation, light adaptation, and chronic vitamin A deficiency, one of the symptoms of which is slow dark adaptation and poor ability to see in poor light. So in a previous lecture we talked about Hippocrates pointing at stars. Okay, that's all for this one.